Hi, I'm Kaylee Livingston, and today I'm going to be talking about the determination of gender, genes versus hormones. So a quick overview. Humans are sexually dimorphic, meaning that males and females are physically distinct. Di meaning two. Genes and hormones play a significant role in sex determination. A human contains 22 pairs of autosomes and 1 pair of sex chromosomes. And if you see in this picture over here, here's all of our 22 autosomes and 1 pair of sex chromosomes. So male and female sex organs, they consist of 3 sets of structures. We have the gonads, the internal genitalia, and the external genitalia. The gonads for males are testes, and the gonads for females are ovaries. Internal genitalia is the accessory glands and ducts for females and males, and the external genitalia is the external reproductive structures for a male versus a female. So sex chromosomes. The 2 sex chromosomes designated are either a X or a Y, both direct development of internal and external sex organs. X chromosome is larger than the Y chromosome, and genetic females are XX and genetic males are XY. And here's the X chromosome and the Y chromosome. Again, X is way bigger than Y. XX is female, XY is male. So sexual differences occur early in development. Differentiation is hard to determine until the 7th week of development. So think of just 7th week, and that's when people tend to go into their doctors and figure out if they're having a boy or a girl. And before they are able to differentiate, the embryo tissues are considered bipotential. Bi meaning 2, and then potential. It could potentially be a male or it could potentially be a female. And the bipotential internal genitalia consists of 2 pairs of accessory ducts called the Wolffian ducts and the Mullerian ducts. As development proceeds, being a male or a female, one of these ducts develops and the other degenerates. So the power of the SRY gene, very important role. So SRI meaning sex determining region of the Y chromosome. And sex determination depends on the presence or the absence of the SRY gene. And if it's present, the SRI gene is present, gonads develop into testes. So if you have an SRI gene, you are going to be a male. And if you do not have the SRI gene, your body is going to develop into a female. So you will have either testes with the presence of SRI gene, or if you do not have the SRI gene, you are going to develop ovaries. And here is what the SRI gene looks like, and obviously it is only in males, not the females. So let's talk about male development. The SRI gene, again, is present, and that's important to remember. And because of the SRI gene is present, testes begin to develop, and testes begin to secrete 3 essential hormones that influence the development of the male internal and external genitalia. So for male hormones, the first one is the anti-Mullerian hormone, and it causes the embryonic Mullerian ducts to regress. So like we said, one of these are going to regress and the other is going to keep proceeding and working up into being a male or a female. And there are androgens, testosterone, and dihydrotestosterone, or DHT. And testosterone converts the Wolffian ducts into male accessory structures. And testosterone controls migration of the testes from the abdomen into the scrotum. And external genitalia is controlled primarily by DHT. So for female development, the SRI gene, let's remember, it's absent. It is not present. If it was present, it would have been a male. But since the SRI gene is absent, female development is going to happen. So the gonad actually develops into ovarian tissue, and the Mullerian ducts develop into the upper portion of the vagina, uterus, and the fallopian tubes. Without testosterone, the Wolffian ducts degenerate, and without the DHT, the external genitalia take on female characteristics. Remember, testosterone and DHT are in males, not females, with the female development. And that is the end of my show.